The process in which the hereditary code carried by the DNA is used by the cell to control protein synthesis has turned out to be quite complex. It involves three different types of slightly different nucleic acids called RNAs and two sequential processes known as transcription and translation. RNA is made up of numerous nucleotides assembled in a similar way to DNA, except that RNA is mostly single-stranded and it uses this ribose sugar that, of course, has an hydroxyl functional group here at the 2'. In addition, RNA does not use thymine, but instead uses uracil, but the other three nitrogenous bases, adenine, guanine, and cytosine, are the same as in DNA. The first step to utilizing the DNA code is the process of transcription. In transcription, the DNA unzips just as if it were going to be replicated, except that instead of DNA polymerase attaching to it, a different enzyme called RNA polymerase attaches to it. Only one side of the DNA is transcribed, and this is ensured by the use of promoters. Promoters allow for the very specific utilization of the DNA. Only areas that that transcription factors bind to will open up away from the histones that they're wrapped around. So the transcription factors have histone acetyltransferase activity, and this allows for the opening up of the DNA and the binding of RNA polymerase. After the RNA polymerase binds, it uses Watson-Crick-based pairing to synthesize an RNA molecule in a very similar manner that, as DNA replication that we've studied before. This RNA molecule is called a messenger RNA. Messenger RNA is informational, very similar to the informational DNA. So the use of messenger RNA is in the sequence of the nucleotides. In addition to messenger RNA, there are other forms of RNA. Probably 24 or so different RNAs have been discovered. We will talk about three different RNA molecules in today's lecture. And then mostly because they represent, because they're used in transcription and translation, but also because they represent the three different possibilities of ribosomal, or I'm sorry, of RNA use. And those are the catalytic, the structural, and the informational. Message RNA has informational content, whereas ribosomal RNA is catalytic and transfer RNA is mostly structural. So ribosomes are made of long strands of RNA as well as proteins. Ribosomes are about 60% by weight RNA. The ribosomes exist in both free pool of ribosomal subunits as well as the ribosomes that are attached to the endoplasmic reticulum and actively translating proteins that are going into the endomembrane system. And so it is not surprising to find out that ribosomal RNA is the most common RNA in the cell. About 80% of the RNA in the cell is ribosomal RNA. the ribosomal RNA is not the only part of the ribosome. There's also a bunch of proteins. So together, there's a large ribosomal subunit and a small ribosomal subunit, and they create active catalytic sites, the E site, the P site, and the A site. They then are able to read the message RNA where a three-letter codon codes for a specific amino acid. There are 64 different combinations of three letters, and there are 20 amino acids. And so this is enough room to code for each and every one of your amino acids. The specificity of which amino acid is going to be inserted due to the mRNA's codon comes from the tRNA, the structural tRNA. And the tRNA has a three-dimensional structure due to intramolecular Watson-Crick base pairing. And so these hydrogen bonds within the RNA create a cloverleaf-like structure that has an anticodon at the anticodon loop, as well as a couple of other loops that allow it to bind to the ribosome and bind to an enzyme that inserts the correct amino acid into onto the tRNA's 3' end, the ACC site. So the specificity of codons comes from these tRNAs because each tRNA has an enzyme that attaches the correct amino acid to the 3' end with a high energy aminoacyl bond to ensure that the anticodon on that tRNA is attached, specifies the correct amino acid. This happens in an ATP-dependent fashion. After the amino acid is bound, then it's just up to the Watson-Crick base pairing of the tRNA against the mRNA to allow for the specificity. However, there's also a reading frame issue. If we were off by one letter or two letters, we would be in the incorrect reading frame, and so we have to ensure that we always start our translation on the correct letter of the message RNA. The way that we do that is by having the start codon AUG, its anticodon tRNA comes preloaded on the small ribosomal subunit. So the small ribosomal subunit comes preloaded with the initiator tRNA, which codes for methionine. When the small ribosomal subunit binds to the mRNA, it scans down until it finds an AUG start codon, and the way that it finds the AUG start codon is by the tRNA Watson-Crick base pairing to it. In this way, the first amino acid of every protein will be a methionine. Once the small ribosomal subunit finds that AUG start codon, it recruits the large ribosomal subunit creating the E, P, and A site. And the tRNA for methionine will be in the P site. The P site is the peptidyl site. The A site is the amino acyl the amino acyl tRNA binding site. The A site I call the acceptor site, and the P site I call the peptide bond site. So when a tRNA comes into the amino acyl tRNA binding site, or the A site, it asks, am I Watson-Crick base pairing to the mRNA or not? If the answer to this is yes, then it is able to stay there for a long enough amount of time that it um, can start forming a bond between the growing polypeptide chain and the amino acid that it brought in with it. If it is not Watson-Crick base pairing, it will just simply leave. After the peptide bond is formed between the growing polypeptide chain and the new amino acid that just came in, then the growing amino acid chain will be shifted to the tRNA that is in the A site. To move it to the P site in a ATP-dependent fashion, the entire ribosome moves down one codon. After shifting its large ribosomal subunit in an ATP-dependent fashion, it then shifts its small ribosomal subunit in an ATP-dependent fashion. Now that the growing polypeptide chain is again in the peptidyl site, we have opened up the amino acyl tRNA binding site, or acceptor site, for a new tRNA to come in and possibly Watson-Crick base pair with the new codon. In this way, single amino acids are added to the growing polypeptide chain, creating the primary structure of the newly synthesized protein. When we get to a UAG, a UGA, or UAA, there is no tRNA that has an anticodon for those codons. Because there is no anticodon containing tRNA, every tRNA that comes into the into the A site and finds a UAA, UAG, or UGA will then leave because it didn't Watson-Crick base pair. If this goes on long enough, then another protein called the release factor will bind to the amino acyl tRNA binding site. The release factor then physically pries apart the large and small ribosomal subunit. This creates a termination. A hydrolysis will happen on the C end, and the entire growing polypeptide chain will be released from the tRNA in the P site. The release factor then pries apart the ribosome, and all of the pieces, the large ribosomal subunit, the small ribosomal subunit, the release factor, and the tRNA go back into the milieu to be used again some other place. The mRNA actually has many ribosomes, or a polyribosome,